Good afternoon. Today we will be presenting our project, AI-assisted classification of microorganism strains on paper-based cultural relics. My name is Allison Wall, and today I'm presenting with my colleagues Chen-Shu Liu and Chongwen Liu. I'm a recent graduate of the UCLA Master of Library and Information Science program. Chen-Shu is joining us from the UCLA Samuel Alley School of Engineering, Department of Bioengineering, and Chongwen is part of the UCLA Getty Interdepartmental Program in the Conservation of Cultural Heritage. So here is an overview of our presentation today. So we'll begin with the background and goals for this project, followed by the project design, next to the project development and results, and finally the conclusion and outlook. So before we begin, here's some background on our project. Our main objective was to create a computer vision-driven microscopic image classification algorithm to classify different microorganism strains on paper-based material using microscopic images. So first, the background and goals of this project. So mold and other microorganisms pose a threat to the longevity of precious cultural objects, not to mention the health of those responsible for their care. Current treatment practices take a broad spectrum approach, treating microorganisms regardless of species. Similarly, while conservators may take precautionary measures through the use of personal protective equipment, or PPE, they're often unaware of the specific health risks associated with a certain strain they are treating. Current mold treatment practices typically take a broad spectrum approach, and you'll hear us use that term throughout this presentation. What we mean by this is that treatments aren't strain or species specific. Conservators will of course assess the object and select the best possible course of treatment, but available treatments treat mold in general without taking into account specific strains of mold. Treatments for mold vary, but typically feature a mechanical cleaning process followed by a chemical one. So first, the object is mechanically treated to remove any visible mold on the surface. This typically consists of dislodging the mold with a brush or a spatula, and then treating it with a HEPA certified vacuum to remove the spores. The conservator will then chemically treat the object both to remove the embedded spores to prevent regrowth and to lighten any stains that are left behind. That way the object is still readable. Chemical treatments vary for paper versus other objects because paper is less tolerant of certain chemicals. Most of the paper conservators we spoke to favored washing the paper and then swabbing it with a diluted ethanol solution. There are also some sources out there that promote the use of an antimicrobial or antifungal solution just to prevent the mold regrowing. Bleaching solutions are also very commonly used to treat the stains after this process is completed. And then after these treatments, the paper is of course dried and dehumidified, and then further monitored to prevent the resurgence of mold. It's important to note that these treatments will vary based on the type of institution. Museums tend to treat objects one-on-one on a case-by-case basis, while libraries tend to deal with a bulk of materials at once, typically in response to some sort of emergency event like a flood that caused widespread mold growth. Furthermore, small institutions without access to the necessary equipment or facilities will often contract out to a vendor conservation company in order to treat mold. So with all that in mind, here are the goals that we focused on in developing this project. So first, we wanted to allow those working with contaminated objects to identify species-specific organisms for the purposes of personal safety and best possible treatment. We also sought to create the opportunity to address biocontaminants while conducting field work, so without access to laboratory equipment. We also wanted to enable institutions to identify microorganisms as quickly as possible, especially in an emergency situation like a flood. Finally, we wanted to develop a means for institutions with limited resources to be able to best remediate mold and to equip institutions to advocate for air quality conditions and the need for personal protective equipment. So with all that in mind, I will pass it over to Chongwen to let us know about the project design.
Thank you, Allison. And knowing that the convenient identification tool would be very helpful for conservators, let's first review what are the current identification methods that has been using in this field and how and why we use computer vision to overcome their drawbacks. So what are the current identification methods for us to identify biodeteriogens, especially on paper-based objects? First thing first, we need to take samples from the moldy objects, and then let's see what microbiology equipped does. These methods can be classified into two general categories: type of more conventional method based on analyzing the phenotype of microorganisms, including first, phylogenetic classification using chromogenic media. The mechanism is that this culture media contains special substrates that can react with microorganisms and react with these microorganisms' enzymatic activities or metabolic capabilities and produce color-producing products. This gives us a means to differentiate different microorganisms based on their specific enzymatic activity and metabolic capabilities. Second are a type of microscopy methods to classify different type of microorganisms based on analyzing their morphology, the shape of their cells or their strings. This is often assisted by fluorescent dyes. And third, there are biomedical techniques focusing on the identification of analyzing the metabolic products such as proteins, amino acids, and lipids. These techniques consist of some chemical methods utilizing these products' chemical reaction with some testing chemical reagents. The most famous one could be ELISA, enzyme-linked immunosorbent assay, which takes advantage of the antibody-antigen reaction to produce visual signals for the target metabolic products. Instrumental methods are also used to analyze the metabolic products. This could include mass spectrometry and chromatography. And there's also a type of more modern instrumental methods that directly analyze the species of a microorganism at the nucleic acid level, enabling the classification of microorganisms based on their specific DNA sequencing. For example, for bacteria, the 16S RNA gene is the standard classification method because this gene is highly specific to different bacterial species, and methods used are primarily PCR, polymerase chain reaction based, which is a technique to amplify a specific segment of DNA followed by different kinds of DNA sequencing methods. So what about the current technologies? These techniques could be problematic for people in our field of conservation to use. From a conservator's point of view, these methods are first invasive because we need to take samples from the already mold-contaminated samples, and they are very delicate. Second, this type of analytical techniques are highly specialized, and this can be only conducted by specialized personnel and labs. Some methods looks easy to use, for example, these microscopy methods, but you will need an expert to help you identify what specific species that is. Because of that, these analytical procedures can be very time consuming, including not only the time you need to analyze the samples. If you are using a culturing method, it might take up to take more than 1 week. And this also includes the time you need to communicate with the labs and with the specialized personnel. And this could be also very expensive, including both instrumental fees and all this consulting and shipment fees. So knowing that these problems with the current identification methods of microorganisms, and they are not very accessible to people in our field, our projects would like to introduce the concept of computer vision in this identification process. So computer vision is basically we enable computer to identify and understand objects and people in digital images, videos, and other visual inputs. In recent years, computer vision has made great progress and can now be considered to be to surpass human vision in these aspects. So first of all, computer vision is exceptional in cases where details are vital. This is to say, computer sees things that we can't see and identify features that we cannot recognize. And also, computer vision is efficient if there's a vast amount of data and they can process a huge amount of data very quickly. And they are able to grab the diagnostic features from a large amount of inputs. And computer vision is also helpful to non-experts and also can be used in various applications. So if they are connected or incorporated with other user-friendly interfaces, like an app on smartphones, we can enable the application in much wider settings. So without eyes and the complicated human visual system, how can computers see things and even surpasses human vision in some aspects? So behind all this is the development of artificial intelligence, as we mentioned in our title, and especially a very specific subfield of artificial intelligence, convolutional neural network, also known as CNN. To make it clearer for us about the terms AI and CNN and some other very popular terms that you might have already encountered somewhere else, like machine learning or deep learning, let's first look at this Venn diagram to get a general understanding. So at the highest level is the concept of AI, artificial intelligence, which refers to smart systems or machines that can carry out tasks that typically require human intelligence. And machine learning is a subfield of AI that instead of giving out fixed rules for machines to perform certain tasks, the algorithms of machine learning can learn from data that we provide to them and make decisions based on patterns observed when given unknown data. However, machine learning algorithms require a certain amount of human intervention when the decision is incorrect. So to enable the machine learn by themselves, deep learning algorithms is developed using a multi-layer structure of algorithm called artificial neural network. In some way, it's actually mimicking the human neural system. Deep learning techniques are able to automatically discover intricate features and reach accurate conclusions without human intervention. And in the end, CNN is a class of artificial neural network that specializes in analyzing data that has a grid-like topology. In other words, it's specialized in analyzing digital media like images, videos, or other things. CNN utilizes a crucial component called convolution layers, which will give a brief description in the next few slides. Next, I'm going to talk about how CNN works, which is almost exactly the same as how we as humans see and recognize objects. So first of all, let's take a look at this picture and ask us some questions. Number one, what do we see in this picture? Obviously, in the front of the picture, we see a camera, we see a zebra, sorry. Maybe we can also recognize the background that it is located in grasslands. And then we might ask, how do we know this is a zebra? In other words, what are the diagnostic features for us to tell that this is a zebra? This probably include, most importantly, the black and the white strips, but also could be the horse shape. And there could also be other features that are less diagnostic, such as this thing has 4 legs, has a hairy tail, and also has a very special ear shape. And these things could also include that the creature is located on some sort of grassland. After identifying these features, we can ask ourselves, why do we know these features that represent this specific thing called a zebra? This is because our human brains have learned from our previous experience, from teachers, documentaries, or even encyclopedias. Just like Encyclopedia Britannica has stated, zebra is a thing that has strikingly black and white strip, and it's a mammal, and it is from the horse family. In the same way, computers do the same thing through algorithm with a multi-layer structure. The first series of convolutional and pooling layers are used to extract the features of the image at the pixel level, like what we just did to point out the black and white strips. In details, a convolution layer consists of just a set of filters that from left to right, the initial layers capture low level and obvious features such as ages or colors, and the deeper layers can capture higher level and more abstract information. This abstract information is difficult to explain, but could be something to describe the zebra as a creature that appears on a grassland environment. Pooling layers don't do anything to the image, but just mathematically assess the convolution layers to reduce the sample size while retaining the most important information. Then the extracted spatial features are then transformed into a single dimension flat vector by a flattened layer to be processed by classification layers, which are usually consist of fully connected layers. The responsibilities of these layers are to make sense of the feature, like what we just did, link these features to a thing called zebra. In the end, the model will give us a prediction in the form of probabilistic distributions, for example, 70% zebra, 20% horse, and 10% dog, which is also easy to understand using our example of human brains. The zebra, if we want to trick you guys, if we want to play with you guys, could be a horse painted in black and white, or a dog we made it to wear a zebra costume. So it is all about probabilities. And in the end, how do we train this model to learn? We provide the model with a huge amount of pictures, and we tell the algorithm that these pictures are zebras, also called the ground truth. As we feed it with more and more ground truth, the algorithm will learn by themselves and then figure out what features to extract or look at and how to make sense of these features. Nowadays, CNN are not constructed from scratch, but through a process called transfer learning. When developers want to develop a model for processing other specific data, for example, medical images as shown in this slide, they will first borrow the convolution layers from some ready-to-use models, such as ResNet 50 or VGG16. These models are trained to identify objects in a widely accepted benchmark database called ImageNet, which contains basically pictures of daily objects. And then they further fine-tune or adjust the model to adapt them into different applications. So instead of training a newly constructed neural network, just like a newborn baby, to learn something, you are training at least a 3 or 4-year-old child that has some basic knowledge of this world. So equipped with this basic understanding of the techniques and technologies, my colleague Chen Shu will then illustrate further how we construct the model.
All right. Thank you, Chuma, for the awesome introduction about how we construct CNNs and how the transfer learning schematic is being applied. So I will go ahead and talk about the project developments and results. So first off was the experimental procedure. We are presented with these moldy materials, which usually presents on ancient Chinese books. As shown here, we can visually see that there are certain places on the book surface that appears to be infested with mold bodies. So that's why we're circling these two regions. One's the red circle and the other one is the orange circle. So for the red circle, we are gently swabbing the surface with a cotton swab, and then this will serve two purposes. The first purpose is let it undergo a series of sequencing techniques to allow us to determine the ground truth. In other words, which specific type of strains we're investigating for. At the same time, the same sample will be used to create mockups for the paper material. And then obviously, because we are dealing with really delicate paper materials, it will be not desirable to directly perform any type of experiments or diagnostics on the original paper. Rather, we are constructing on Chinese Xuan paper or a type of ancient Chinese rice paper as a mockup for the book materials. So we'll create these mockups, and then the mockups will be taken under the microscopes, the compound microscope, as well as the foam-attached microscope to get the image of the moldy surface underneath. So those images, together with the ground truths we've determined through the sequencing techniques, will be fed into the a deep learning algorithm in order for it to train itself. And then we'll end up with a deep learning classification model ready to use to perform this type of microorganism classification tasks. And that is the training process. Then we're ready to dispatch this deep learning classification model onto any type of device that can just process any image data. So in this case, we can use a phone or a computer or a tablet, whichever one that works in that scenario. And then we'll take a picture of another location where the algorithm has not seen it before. And then we'll also determine the ground truths. Using the phone, it will just non-invasively take a photo of that region. And then we can use the ground truths as well as the predicted label to determine if the model can correctly classify the type of species infesting on the surface. And this is called the testing phase. So here are the photos that we got. So on the left, there are the 4 books that we obtained the microorganism samples from. And then on the right, there are the 4 mold species or microorganism species that we investigated. So these are the microscopic images that we collected from the ancient book samples. So 2 types of microscopic images are collected, and one is from the compound microscope, which is the most commonly seen apparatus around the lab. And then the other one is a foam-attached microscope, which we are trying to see if having these lens attaching to the phone camera will allow us to directly obtain a microscopic image. And then we want to simulate this scenario where the compound microscope is not readily accessible, which can apply this algorithm to a wider application realm. So having all these images, we need to further feed that into the algorithm. So this pipeline is showcasing how we collected the images. So first, we gently swap the book surface to obtain the moldy parts or the mold bodies. And then we let it undergo a series of zero dilutions to purify and isolate the microorganism strain. And then we gently spray the culturing solution together with the microorganism body onto the mock-up papers that we pre-constructed. And then after letting it to grow for a bit of time, maybe around a week, we'll take a microscopic image under both the foam-attached microscope as well as the compound microscope. So obtaining the images is not enough. We need to further introduce more variations to the image in order for the algorithms to have a more confident prediction accuracy. So that being said, in order to introduce more variations to the images, we are using an image augmentation pipeline. So first, we're presented with this original image, which is 2000 by 2000 pixels wide and height. And then we're randomly cropping segments within. So for example, we're cropping 1000 by 1000 smaller segments from the original image. And then this is not enough. We need to further augment the variations within the images. That's why we introduce a rotation to the images. So the type of rotation that we use are by multiples of 45 degrees. So that's why we are seeing these the squares and also octagon shape images. So having multiplied the original image into 7 different variations will allow us to have more variations in the image dataset and further improves the model's confidence when they are predicting for other scenarios. So after this image augmentation task, we're ending up with all these images. So for the raw total for all these the 4 strains of microorganisms, we can see it's clearly not enough. So after the augmentation, we have more than enough images for training the model and evaluating the model. So now moving on to the modeling pipeline. So as Shulman mentioned, we are using a CNN network, which stands for convolutional neural network. So convolutional neural network will be presented with a image input. So the image is by type RGB, or having 3 channels, red, green, and blue. So the image will be put into a sequences of pipeline called feature extraction. So after that, we should be able to end up with several feature maps to describe the type of components being described on the images. After that, we'll let it pass through a global average polling layer, which is sort of aggregating all the feature maps we have obtained from the image through this feature extraction pipeline. And using this aggregated feature maps, we can further pass down them into a classification layer, which ends up with a probabilistic distribution about which type of strain the image is depicting. Further, since we're not developing a algorithm all from scratch, we're using a transfer learning schematic. We have experimented with different type of transfer learning pipelines. Namely non-fine-tune, partial fine-tune, and full fine-tune. By no fine-tune, we mean that the pre-trained network, the convolution layers or the feature extraction layers being transferred from previously trained models are not designed to be trainable in our case. So whatever is learned from the previous cases will be frozen and will not be trainable. And so those knowledge will not be altered, except for the last classification layer, which will adhere to our scenario, which is classifying the 4 type of microorganisms. So this is for the no fine-tune case. And then for the partial fine-tune, we're just partially unfreezing, letting part of the transferred model to be trainable, to adapt to the new scenario, and letting the other half to be non-trainable and retaining the original knowledge that it has. So usually we just unfreeze the top layers or the layers that are on the left. So those layers, since they are depicting features that are usually lower level or easier to interpret, such as like an edge or a color, so we are freezing those information. And then in order for the model to adapt to our new scenario, we need to let it to learn some higher features from our new images. So that's why we are unfreezing or letting the latter layers or the layers on the right to be trainable. And then the last one is the full fine-tune, which is basically just tuning the model as a whole. So whatever is being transferred will be altogether trainable. So the only thing that is kept unchanged is the structure being transferred from the previous models. So now comes to the classification model performance. In order to showcase the model is being competent enough to predict what type of microorganism is infesting the surface of the paper, we're using this confusion matrix. So this confusion matrix contains a horizontal axis and then the vertical axis. By the vertical axis, we are denoting the ground truth or the true label of the image being shown. And the horizontal axis is actually showing the predicted label, which coming from the algorithm that produces the classification results. For example, if we look at CLA or cladoporism, it shows that there are 113 images being correctly classified. So whichever square that appears to be darker and the number within it appears to be higher shows that the accuracy is higher. So to put it in numbers, we can see that for the 4 strains, all of them have over 85% accuracy, and even 2 has over 99% accuracy, proving that this computer vision structure can be applied or can be transferred into the conservation diagnostic field. So in order to further showcase that our model is actually learning the features from the mold bodies instead of something that is confounding, we are using a feature map to showcase the competence of this model. So a feature map refers to the output of a particular layer from the network, and it kind of represents like which part of the image that the deep learning model deems important for the classification task. So by visualizing the feature maps, we should be able to see that if the deep learning, deep convolution neural network model is actually learning the things that we want them to learn. In other words, the mold bodies and the morphologies of the microorganisms themselves, instead of like something on the background. So in our case, since we are dealing with the mold growing on a cellulose fibers or paper structure, and paper are usually made up of cellulose, the background or the cellulose fibers can usually act as a confounding variable. So that's why we are seeing this feature map. So if we compare the image on the left, which is the original image, and the one on the right, which is a feature map, we can see that for the mold bodies or the black dots, those are being highlighted. And then for the cellulose fibers on the background, they are not being highlighted. So if I were to overlay these two images on top of each other, we can see that the mold bodies are being deemed important by the model for the classification task. On the other hand, for the cellulose fibrous background on the background, those are not being considered. In other words, the model is just using the morphological information from the image about the microorganism bodies instead of something on the background, such as the cellulose fibers, proving that our model is actually learning the morphologies of the colonies and the mold body individuals instead of like something like the cellulose fibers. So after that, I would just pass on to Allison to further talk about some concluding remarks.
All right, thank you for that. So to summarize, we would like to discuss the progress of this project as well as its outlook. This project integrated computer vision in the identification of microorganisms on paper-based relics and demonstrated its successful application on 4 microorganism species on Schuin paper samples with high accuracy. Compared to conventional identification methods, the application of this model has the potential to be non-invasive, accessible, fast, and low cost. In addition to being able to identify strains of mold for the purposes of treatment and personal health, we discovered some additional potential uses of the software after discussing our project with several paper conservators working in different types of institutions. One paper conservator with a museum background pointed out the software's potential for provenance identification. Eventually, it could possibly be used to link objects together or to certain locations or geographical events based on the species of mold identified. Another conservator working as a vendor for a conservation company said she could see it being useful for insurance purposes. She stated that the software could help her to report more thoroughly on object conditions, for example, which species of mold was identified, whether it was more difficult to treat, grows faster, is more toxic, etc. So this could potentially be useful to fully communicate the extent of mold damage. And some additional benefits. So another conservator we spoke to works in a large university library and mentioned that she could see this being useful for advocacy purposes. So she herself was experiencing some allergy and respiratory issues, and mold was a very common issue in her library. But her university had several years prior had the air quality tested, and it came back that the air was fine. So she said this would be useful to report thoroughly to the administration which species of mold was showing up, how it affected people's health, and why it should be addressed. And finally, an additional benefit that we foresee with this software is resource allocation. This would help all institutions, but especially small institutions with limited resources. These organizations could best utilize limited resources by treating specific strains of microorganisms rather than general broad spectrum treatments. So all in all, our goal for this project is to enable the development of strain-specific treatments that, rather than more general treatments that may be less effective. This is a non-exhaustive list, of course, but just some examples of how we see this technology being applied within different types of organizations in the future. And now I'll hand it over to Chen Xu to discuss project outlooks.
All right, thank you, Allison. Some future outlooks and possible improvements of our model currently is since we're developing the current model as identifying each strain independently from a single image. However, in real life scenarios, there might be cases where there are multiple strains presented on the same image. So one possible improvement is using a multi-label classification, which is able to handle like multiple strains on the same image to replace the current multi-class classification model. And further, we're developing a mobile app for phones for conservators to use. So this will be a more applicable scenario, considering that not all the situations has a compound microscope at hand. So that's why we're having this mobile phone application ready to use, having this deep learning model embedded into the phone in order for the conservator to just directly take a picture of the moldy surface of the book, and then it can directly produce a classification result. And then we're also envisioning a AI plus conservation ecosystem. So that being said, we're considering creating an image database that enforces resource sharing, which can brings in much variety of image sources. And not only can it be like images, it can also be like conservation practices and etc. So having this image database or database overall would allow us to train more robust models, and at the same time will allow us to explore different type of AI plus conservation type of algorithms. So one thing that we think about is having a knowledge base constructed to sort of analyze the possible or the best way for a conservation practice, considering a certain type of situation. And we can take advantage of the currently most popular idea of large language models, such as the ChatGPT, to create a recommendation system with this database and the knowledge base to provide the conservators the best practice to use when they are dealing with a specific type of situation. That being said, this project will not be possible without our fellow contributors back in China. Special thanks to Professor Songbin Ben from Liaoning University and graduate students Yilin Hao, Xianchao Li, Qingxia Meng, Junqiang Geng. So it will not be possible without their image collection, without the culturing of the mold bodies, as well as culturing the mock-ups in order to mimic the paper backgrounds for our to process the images. And then we also want to thank all the connections we had earlier during the project development process. Professor Allen, Tamia, Marika, Erin, Maria, Christina, and also the Liaoning University Library Special Collections. They during our contact with them, they provided us with such good feedbacks and then allowed us to further proceed with our project and improve on our current status. So I want to say thank you to all the person we have connected with, and that will be all for our presentation.